how much is the strength of the stimulus, the strength of the response should also correlate with that. And for that, your inhibitory neurotransmitters are really playing a very important role. So GABA is the main inhibitory neurotransmitter in the central nervous system. So occurring in 30 to 40% of all the synapses. So it is located in high concentration in the substantia nigra and globus pallidus nuclei of the basal ganglia. It is also present in the hypothalamus, periaqueductal gray matter, as well as your hippocampus. So the concentration of GABA within the brain is 200 to 1,000 times greater than that of the monoamines like your catecholamines or your acetyl-CoA, which are important excitatory neurotransmitters which are there. So from this particular thing, this particular fact also, we understand that how important inhibitory neurotransmitters are, how important your inhibition is. Okay, so gamma aminobutyric, that is your GABA, is created from glutamate. So by your enzyme glutamate decarboxylase. Okay, so let me just show you. This is the structure of GABA, that is gamma aminobutyric. This is the molecular formula C4H9 NO2. So it's a deuterion. So as any other amino acid, so it is with having a protonated carboxyl group and deprotonated carboxyl group and protonated amino group. So this is how your synthesis of GABA will take place. So using your glutamate decarboxylase, glutamate is going to be converted. So because of carboxylase, the CO2 is carboxyl group is removed, and it will become your gamma amino butyrate, that is your GABA. And there are different transaminases which will lead to the breakdown of this GABA. So these are usually being present in the mitochondria as mitochondrial enzymes, and is responsible for the degradation of the GABA, which is also one of the important factors which needs to be taken into account, as we have learned that every neurotransmitter should have an enzyme which may be able to degrade it, and also should have transporters which would be able to take up the neurotransmitters back into the neurons from the synaptic cleft, right? So you have GABA glutamate transaminase, which is responsible for breaking down your GABA into succinate semialdehyde, which would be further broken down by your succinate semialdehyde dehydrogenase into your succinate, okay, which could be used for different metabolic reactions further. So storage, you have newly synthesized GABA is stored in synaptic vesicle by means of your vesicular transporters, which are specific for this, and they are stored at postsynaptic terminal until your action potential is released. So that's that's as with any other neurotransmitter. So stored GABA released into synaptic cleft, stimulated by depolarization of the presynaptic neurons. So GABA diffuses across the cleft to target receptors. But here, the action of the GABA is terminated by the reuptake of the GABA by presynaptic nerve terminals as well as your glial cells. So you can see over here the entire procedure. So whenever your glutamine, it can be it is converted into glutaminase, or using your glutaminase, it will convert into glutamate, and it will get converted into GABA with the help of your glutamic acid decarboxylase, and then getting transported into the vesicles using your vesicular transporters. Yes, and then this vesicle containing your transporters, they will reach to the presynaptic zone where using your exocytosis, it will be released. So again, you have GAT, that is your GABA transporters, which are present onto the these you have it on to the membranes, which will help in taking up. They would be also helped in taking up to your glial cells. So these are your glial cells, which will also help in taking up of GABA from the this synaptic cleft. and further converted into your glutamine using your glutamine synthetase and transported back to the cells, to your neuron cells, so that it can be reused for the formation of the GABA. Okay, and then here at the postsynaptic neuron, again, you have the receptors which can bind to your these neurotransmitters, that is your GABA, and which are associated with channels and lead to. So here we will be learning that instead of leading to your depolarization, because it is an inhibitory neurotransmitter, then it will lead to your hyperpolarization. Yes, GABA will lead to hyperpolarization instead of depolarization, right? So receptors, there are two types of receptors, GABA A and GABA B. So again, as we have seen in case of acetylcholine, also they will have different pharmacological and biochemical properties. So GABA A is ionotropic. That means it is connected or it is associated with ion channels. And the GABA B is metabotropic. That means it is associated with different secondary messengers and is involved in this complete signal transduction. So let us move forward and understand about these two receptors in detail. So as we were talking about the synapses which use GABA, they are referred to as your GABAergic synapses, and it acts as we have seen that neurotransmitters would be reuptaken, and GABA A and GABA B would be most analyzed of the 5 types of your GABA receptors. There are other receptors also, but we don't have much knowledge about that. But the most importantly characterized ones are your GABA A and GABA B. And whenever there is any dysfunction of these receptors, a lot of many disorders are associated with that. So dysfunction with this inhibitory neurotransmitter can cause seizures, it can cause a lot of anxiety disorders. Okay. So as we have said that GABA A is ionotropic receptor and is ligand-gated ion channel. Ligand-gated, that means because of the binding of GABA to these receptors, the opening of the channels will take place. Okay. So the ligand is your GABA, which is your inhibitory neurotransmitter. So upon activation, so when your GABA goes and binds to this GABA A receptor, it selectively conducts your chlorine minus through its pores. That means it will open up the channels for chlorine ions. Kindly note this, chlorine ion, Cl minus. Okay, for most of the excitatory neurotransmitters, it is your potassium and sodium ions. But in case of your any inhibitory neurotransmitters, it will start conducting, it will start allowing your Cl minus ions to come inside, and that will lead to the hyperpolarization of the neuron. This particular part has to be noted by all of you. And this causes an inhibitory effect on the neurotransmission by diminishing the chance of a successful action potential occurring, because if there is hyperpolarization, then you require more number of your excitatory neurotransmitters to make it depolarize, which becomes more and more difficult. So until and unless the stimulus is very, very strong or the action potential is very strong, so that this more hyperpolarized neurons are depolarized, until and unless that is being done, you will not be able to, or the action potential will not occur in these neurons. Okay, so that is what is being said as your fine tuning of these neurons. Okay. So the active site of your GABA A receptor is the binding site for GABA, and various different drugs also bind to this. Like you have muscimol, you have gabapentin, you have baculin. So some of these will act as your there are different molecules. We have talked about your molecules that will mimic the action. Quickly tell me what are the molecules that mimic the action called as, and what are the molecules that inhibit the action called as. Mimic the action wala pehle bolo, which is what are the molecules that mimic the action of your neurotransmitters called as? This is a revision quickly. Agonist, very good. And yes, uh, no, they are called as your agonist molecules, and the ones which are inhibiting, they are called as your inhibitory, right? So there are many drugs which can act as your agonist and there are many poisons or drugs which can act as your antagonist as such. So if there is an agonist, yes, that it will help in the inhibitory thing. It will help in inhibition of the action potential. Whereas when you have antagonist in this case, then it will lead to more hyperactivity because antagonist will not allow the GABA to do its action, and it will lead to the hyperactivity. So the protein also contains a number of different allosteric binding sites. Yes, what are allosteric binding sites? These are regulatory sites. Most of the times the enzymes or the proteins, they have two sites. One is active site where your substrate goes and binds, so the ligand goes and binds, and you have an allosteric site which is mostly for modulating the activity or regulating the activity of those proteins or your receptors or your enzymes, what you can say. So there are various different drugs which can bind to this allosteric site, which will further help in regulating the activity of these receptors. So like you have your benzodiazepines, then you have non-benzodiazepines, you have barbiturates, you have ethanol, that's your alcohol, then you have neuroactive steroids, you have inhaled anesthetics, you have picrotoxin, and all these which will lead to the inhibitions of these. So these benzodiazepines do not bind to the same receptor site on the protein complex as your GABA, so they are binding to other sites. So, but binding to distinct binding sites situated at the interface between alpha and gamma subunits. And majority of your gamma subunits, those having your alpha 1, alpha 2, alpha 3, and alpha 5 subunits, are sensitive to benzodiazepines. There exists a minority of your GABA A receptors which may have your alpha 4 or alpha 6 subunits which are insensitive to this particular class. That means if I show you, this is your GABA A receptor. So there are different types of your subunits which are there. Okay, so this is the place. Okay, so where you see this round yellow space which you can see. So this is the space. Just give me 1 second. I'll annotate and show it to you. So this is the space you can see over here, okay, which can bind to GABA. So this is your active site, whereas your this benzodiazepines which are there, they are specifically binding to these ones, okay. So now again, depending upon, so this is the interface of your alpha and gamma which are there. So again, each of these subunits, so alpha, there are different types, alpha 1 to alpha 6, 6 different types are there, okay. So depending, there are different types of your GABA A, depending upon what structural subunits are coming together. Like beta will have beta 1, 2, 3. Gamma will have gamma 1, 2, 3, or you can also have your this delta epsilon pi or theta. Okay, so different subunits can come together to give rise to different types of your GABA A receptors. So that's what was being talked about, that depending upon if a particular GABA A receptor is having your alpha 1, 2, 3, or 5, okay, then they will be able to respond to this benzodiazepine and get affected by it. Whereas if apart from if there is alpha 4 or alpha 6 subunit present in these receptors, then they are insensitive to this particular drug. So instead of sensitive to other classes of GABAergic drugs such as steroids or ethanol, so these receptors will not be responsive to benzodiazepines, but definitely would be sensitive to neurosteroids and your ethanol, that is your alcohol. Okay, so that's how different. That's how your GABA A receptor will look like. So if you have just an overview from top, you can see this is how your receptor is going to look like. So at the interface of your alpha and beta, you have the binding site for GABA, and at the interface of alpha and gamma receptors, you have the site for the benzodiazepines to bind. Okay, so that's how you can see over here. So different types of your benzodiazepine sites. So some of them will act as agonists, some of them will act as antagonist. There are different sites for binding of barbiturates. That is your different types of your drugs which are there. Then there are different sites for anesthetics to bind. Now, can you people just think and tell me what would the anesthetics be? Like what would be the anesthetics for your GABA receptors? Would it be an agonist or would it be an antagonist? Think and then answer. Anesthetics will be an agonist for GABA receptors, or should it be an antagonist for your gamma receptors? Yes, quickly. A quick answer from everyone. Think and answer. And I want everyone to answer, please. मैम कुछ अर्जेंट है मैं लेक्चर में हाँ मैं करती हूँ लेक्चर के बाद करती हूँ Yes, students, answer quickly. Just use your brains. You should be able to understand. Can you repeat the question? Okay, my question is, what effect will your anesthetics have on GABA receptors? Okay, anesthetics. We have. I'm showing you that you have many anesthetics which have binding sites for your GABA receptor, right? So when they bind, will they act as an agonist or will they act as an agonist, antagonist? Sorry. I want answer from everyone. 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 So apart from Nidhi, everyone is saying agonist. Yes, can anyone explain me why agonist? Okay, Pranali is also saying antagonist. So can anyone? Shrutika, unmute. Shrutika, unmute. Yes, Shrutika. हाँ अनम्यूट करो एंड टेल मी ऐसे कैसे माइक नहीं काम करता है फोन से ही तो बात कर रहे हो फोन पे फोन का माइक नहीं काम करता है ओके टेल मी श्रुतिका व्हाई इट शुड बी एन एगोनिस्ट एंड व्हाई नॉट एन एंटागोनिस्ट एक्सप्लेन Nidhi and Pranali will explain me why it is an antagonist and why not an agonist. Quickly, everyone, be prepared with your answers. Yes, I want quick answers. Yes, Soumya has written that GABA inhibits the activity of neurons and hence if given an anesthetic drugs, it will not reduce the activity further as the drugs will enhance the activity are called as agonist. Yes. Yes, and anesthetics का role क्या होता है कि your body is not responsive, right? You uh, every action potential is going to be it's not taking place. You can't feel anything. You can't feel the pain or anything, right? So definitely it has to be an agonist because inhibitory ones, numbing effect, and also inhibitory ones are the ones which will enhance this effect. If you are trying to enhance their activity, then your body would be more unresponsive, right? So that is the that's how your neurotransmitters will work. So even barbiturate, these drugs, okay, they slow down our body. If you know that these drugs, they will slow down our system. Alcohol will slow down our system. A person's, cognition levels, understanding, everything slows down. Why? Because these drugs will go and bind to these GABA receptors and act as an agonist. It will enhance its activity, and that's how our body, our muscles, and everything they are not in our most of our it becomes slow. It is not in our controls. Okay, so everything will act as an agonist. Okay, so that was about your GABA A receptor. Then going forward, we have GABA B receptor, which are metabotropic transmembrane receptors for your GABA, and they are linked to your G proteins and also to your potassium channels. So what they will do is change the potassium concentration and hyperpolarize the cells at the end of the action potential. We have seen that the your potassium concentrations have to be more, and that's how using your different combinations of potassium and your sodium channels, we have seen that the polarized state is created, right? So here they will change this potassium concentration in such a way that the hyperpolarization will take place, and this GABA B receptors are found in the central as well as your peripheral nervous system. So they will opening up of they will result in the opening up of your potassium channels, which will bring neurons closer to the equilibrium potential of K plus hyperpolarizing the neuron. That means the amount of K plus uh, whatever your ions are there, that is going to be equilibrium. That means same inside the cell and outside the cell, which should have been uh, more inside the cell and less outside the cell. So this prevents the sodium channels from opening up because this exchange has to take place, right? So sodium channels will not open up and the action potential will not take place. Okay, so it will stop the neurotransmission, the entire action potential will stop, and so there will not be opening up of your calcium channels at the presynaptic zones, and thus GABA receptors are considered inhibitory receptors, or this GABA B receptors. So they will reduce the activity of adenyl cyclase and decrease the cell's conductance to calcium plus. And we have seen that calcium plus is very much involved in the releasing of your vesicles in your presynaptic zone, right? So if the vesicles are not being released, the neurotransmitters will not release, and the entire action potential is going to be stopped. So these GABA B receptors are involved in behavioral actions of your mainly your ethanol, your gamma hydroxybutyric acid, and also in pain. Okay, so these receptors in the pain, whatever your neurons which are involved, or the cortex which is involved in the pain sensation, there your this GABA B receptors are playing very important role. So this is how your GABA B receptors will look like. Okay, so they are heterodimers. You can see. So two GABA B subunits have been cloned, that is B1 and B2, and B1 subunit has GABA binding region. So you can see over here, this is your B1, where your GABA will come and bind this red dot, which you can see, that is the GABA, while your B2 subunit which is there, it is most of it is not having any binding site for GABA, but it is having interactive sites for your G protein. So it would be associated with the G proteins which is present in the transmembrane. Okay, so the major action, as we have seen, is to decrease the calcium conductance, calcium plus 2 conductance, and increase the potassium plus conductance. Okay, so that's the effect that these GABA B receptors are going to have. Okay, so there are other interacting proteins that are there, like your adenosine molecules, trafficking molecules. There are various transcription factors which are affected by these receptors. You don't have to go in much of these details, but these are the things that you need to remember for your GABA B receptors. Yes, I hope the gamma receptors or your GABA is clear to all of you. Okay.